By the end of this video, you'll know what TB-500 is, how it works, what the evidence suggests, what the risks are, and how it compares to BPC-157. TB-500 is the thymosin beta-4 peptide fragment made up of the amino acids LKKTETQ. Some papers also mention a C version of that, which is just the acetylated version of that fragment. So full thymosin beta-4, it's a much larger 43 amino acid peptide, and TB-500 is related to that biology in that it is a 7 amino acid fragment of the larger thymosin beta-4 molecule. And one of its main known jobs, which we'll get to later, is helping control actin, which is a structural protein cells use to move and repair tissue. And the gene that codes for thymosin beta-4 is TMS-B4X, and that just stands for thymosin beta-4 X-linked. TB-500 is typically described as thymosin beta-4's active actin binding region. So the best example I can give of TB-500 is it's like taking the most important phrase out of a larger paragraph. And there's also different product names for things that contain thymosin beta-4 or TB-500. You might see names in some of the studies such as timbitasin, RGN-259, RGN-137, 352, and NL-005, and their product development contexts with specific routes, tissues, and endpoints attached to them. And also one more thing I should mention is that there's quite a bit more research on thymosin beta-4 than there is on TB-500, but today I'll try to break down the differences. So let's talk about the proposed mechanism for TB-500 and how it could work. So the full thymosin beta-4 peptide has the clearest evidence for binding G-actin. And G-actin is just the soluble form of actin that cells use to remodel their shape and move. So TB-500 could also theoretically bind G-actin because it's based on that active region, which sits right in the actin binding portion of thymosin beta-4. So the most cautious speculation here is that TB-500 could theoretically help repair by nudging cytoskeletal remodeling and repair cell movement, but not necessarily by acting through a well-defined TB-500 receptor. So actin, as I mentioned previously, is the movement hardware of a cell. So when a cell crawls, it pushes its front edge out, it goes forward, it grabs the matrix of the tissue through things called integrins and focal adhesions, and it pulls the cell body along, and then it releases the back of the cell. So full-length thymosin beta-4 is tied to that system through this G-actin binding that we've mentioned, and TB-500 is related to that mechanism. So the G-actin to F-actin drawing here I have for you is the center of this mechanism. So G-actin is the loose monomer, F-actin is the filament. At the leading edge of a moving cell, these actin monomers get added to growing filament ends, especially barbed ends. And that polymerization, it physically pushes the membrane forward, and that's the first downstream effect. So we get protrusion, and you can see it in this sketch of the cell here. So we have these different lamellipodia coming off of the cell. Those are those green protrusions, and you can see the cytoskeleton poking out there, and that's that F-actin chain that's progressively getting these G-actin monomers added to it. And the direct binding piece is this. The full-length thymosin beta-4 binds G-actin in a way that blocks that actin monomer from joining a filament. So structural work shows thymosin beta-4 contacting the barbed and pointed faces of G-actin. So thymosin beta-4 is not simply activating actin, it's actually holding and regulating the monomer pool. And then profilin here, let's add it to the mechanism. So profilin, it can take actin monomers into a polymerization ready state and support the barbed end filament growth. And then downstream of this, we get a repair cell. It senses a wound signal. So let's say this cell, it senses a signal, and then it releases or hands off actin monomers near the leading edge, and it builds F-actin. It pushes out this lamellipodium that I would mention earlier. It sticks through integrins, and it contracts through actomyosin, and then it lets go at the rear of the cell. And then when we repeat that cycle enough times, the cell actually moves into the injury area. So let's look at the direct evidence for TB-500, not thymosin beta-4. And this is a little bit narrower evidence base than thymosin beta-4. And the direct papers are mostly about, identity, detection, metabolism, as well as a few preclinical models. So the first study here is a 2012 paper that characterized the acetylated version of TB-500. And this paper just found that it was the 17th through 23rd amino acid that was the fragment of thymosin beta-4 that was considered TB-500. And so this was just identifying the molecule. And there's also a study on doping in horse doping. So this was a 2012 paper, and they developed methods to detect TB-500 and its metabolites in horse urine and plasma. And they found that TB-500, it doesn't just remain as one intact molecule in the body, it can actually be metabolized into shorter peptide fragments. And this is important here because these are just analytics, so this is not exactly saying how it works in the body. And so there's a 2024 metabolism paper. It looked at TB-500 and its metabolites in human serum or enzyme systems, rat urine, as well as fibroblast assays. And they found that TB-500 may actually act less like one intact molecule with one clean target and more like something called a profragment. It just means that a short starting piece gets broken down into smaller pieces that may actually be the real active forms. And in this study, the standout candidate was acetylated LKKTE, which actually appeared to improve fibroblast movement in a wound healing assay, while the parent TB-500 itself was not the clear active hit here. And so that's actually a really useful nuance. So the parent here might actually not be the specific thing that is very, very active in the body. But again, this is just one study. So there's the 2025 corneal hydrogel study. This is probably the cleanest direct preclinical example using the TB-500 associated fragments. Okay, that's the LKKTETQ. That's the full TB-500 molecule. And this was not using thymosin beta-4. And they used this local hydrogel model for corneal injury repair. And the hydrogel delivery system here was designed to respond to alkaline phosphatase, or ALP for short, and it's an enzyme signal that's present at the injured corneal surface. So the experiment was not like, let's just give TB-500 and see what happens. It was, let's place TB-500 in a material that activates locally and temporally at the wound site. And the main cell readouts for the study were specific to human corneal epithelial cells. So the authors looked at these corneal epithelial cell migration, proliferation, and tight junction recovery. These are all important because the corneal surface, it has to close quickly and rebuild a barrier that keeps the eye protected and optically clear. And they also had this stromal fibroblast data, and it added a second corneal cell type. So they tested this to report changes in cytoskeletal organization and differentiation-related behavior, and this was also another thing they added. So they also tested the hydrogel beyond the cell culture using an animal corneal alkali burn injury model. And I just want to be clear that this was only this last part of their study that used the animal model. In that model, the TB-500 hydrogel accelerated epithelial regeneration, reduced inflammatory signals, and improved barrier recovery. And so overall, that was kind of their main takeaway from their corneal study here. And this just makes the result a bit stronger than a Petri dish only experiment. So that's the direct TB-500 evidence. And the summary is that the bucket for direct evidence is pretty small, and it's quite a bit smaller than the thymosin beta-4 literature here. So now for the human data. This was a phase 2 study with 72 participants. And here, it was actually using thymosin beta-4 and not TB-500. So it's comparing 0.1% thymosin beta-4 eye solution with a vehicle. Vehicle is just like a placebo. And they were looking at ocular discomfort and corneal staining in a controlled adverse environment. And the result was mixed, and that's the point here. So the primary endpoints, they did not significantly differ at day 29, but some secondary signs and symptoms improved, and there were no adverse events observed in that report. The severe dry eye signal was encouraging, but it was quite small. So there was only 9 patients, and the study also reported symptom and staining improvements with thymosin beta-4 eye drops. And neurotrophic keratopathy is more of a wound-like example because the endpoint is corneal epithelial defect healing. And in this endpoint, they found complete healing with thymosin beta-4. And yeah, so this is interesting. So 6 of the 10 healed on RGN-259 versus 1 of 8 on placebo at 4 weeks, with p-values equaling 0.0656. So when I say RGN-259, that's just a product form of thymosin beta-4. And for skin results, a phase 2 venous stasis ulcer study with topical thymosin beta-4, that's what it was, and the safety across doses was reported as acceptable and comparable to placebo, and the 0.03% signal suggested possible healing acceleration here. And the heart evidence, it uses thymosin beta-4 as well, not the TB-500 fragment, and they looked at percutaneous intervention. It's an emergency non-surgical procedure typically used to restore blood flow to the heart during a heart attack. And so they wanted to look at what happened here. And these are like acute heart attack patients. So this is important. There's 96 patients, and the study was 90 days. It was randomized, and the result was split. So an early first dose subgroup had a reduced heart attack area signal, but the full 96 patient analysis did not show significant heart attack or infarct area differences. So let's compare TB-500 with BPC-157 because people often talk about these two compounds together. And the key here is not like which one's better. We're just going to compare and look at both of them here and where they might overlap or be different. So BPC-157, it's usually described as a synthetic 15 amino acid gastric peptide. TB-500, on the other hand, is the 7 fragment from thymosin beta-4. It's not the full thymosin beta-4 peptide. And the evidence buckets, they're uneven in different ways. So BPC-157 has this broad repair and orthopedic preclinical literature, but thin human musculoskeletal evidence, whereas TB-500 has a tighter fragment identity. It's much smaller, and some direct preclinical signals, but much of the human evidence belongs to thymosin beta-4 products, which has way more research than TB-500 does. And mechanistically, BPC-157 is often talked about through vascular repair signaling, so pathways like the VEGFR2 pathway, ACT, eNOS, and nitric oxide. And BPC-157 also has a repair cell lane, so tendon cell work points to migration, things like survival under stress, F-actin changes, and FAC-paxillin signaling. So the broader BPC mechanism also keeps coming back to barrier and cytoprotection models, especially gut and wound settings here. The TB-500 side, though, it starts in a bit of a different place. So it starts here with thymosin beta-4 biology. So as we described earlier, it starts with this actin handling, the cytoskeletal rearrangement, cell migration, endothelial behavior, angiogenesis, and repair environment remodeling. So this is more of an actin and migration mechanism than a BPC style vascular or barrier mechanism. So the shared zone, though, here is cell movement, as well as vessel behavior, things like inflammation timing, as well as matrix remodeling. So BPC-157 maps more naturally to vascular, repair cell, matrix, and barrier signaling, whereas TB-500 maps more naturally to actin migration and thymosin beta-4 vascular-related repair biology. So they do have quite a few similarities, but their initial mechanism is different. And there's a number of different risks with TB-500 and or thymosin beta-4, but the largest risk would be the potential increased rate of angiogenesis feeding a pre-existing cancerous growth. So the concern is that tumors also use angiogenesis. So once a small tumor has enough growth signals, new vessels can help it get oxygen, nutrients, as well as a route for expansion. So this is the largest risk. So there are a lot of other risks associated with it. The WADA has released a statement saying it's prohibited at all times, as well as there's a lot of unknowns and things we just don't know about TB-500. So we know what TB-500 is for its fragment identity, and we do have direct preclinical TB-500 signals, and we also have specific human thymosin beta-4 product data, so that's all great. But for what's plausible and unknown, so repair environment biology is plausible, and the adjacent thymosin beta 4 human product signals are like legitimate, but they are still narrow. And things like direct human TB-500 recovery benefits, what the ideal route is, what the long-term safety risks are, as well as BPC-157 combination effects, we still need trials for those. So that's it for the video. Thank you so much for watching, and I hope you have a great rest of your day.